Hello all. My name is Maria Lorena Ginga, and I'm so happy to be here in front of you because it's my first experience at the conference. I'm part of biotechnology and bioanalysis team, and our work involved the development of a portable and miniaturized electrochemical tools for rapid and simple determination of nitrate in soil. First of all, I will highlight the importance of nitrate and the advantages of using nanomaterials in sensing applications. I'll present after the design and development of these sensors, the morphostructural and the electrochemical characterization. And I'll finish by presenting the detection of nitrate in real samples. Why nitrate? Nitrate represents an essential plant nutrient which takes part of the plant nitrogen cycle. It is found in small amounts in aquaculture systems, but its accumulation in soil can lead to eutrophication bodies. The table presented here presents the toxic nitrate levels at different pH values. And these concentrations are so important because an excess of nitrogen can lead to acid rains, greenhouse effects, or health hazards, especially to infants. The nitrification process involves more steps, but the key one is the oxidation of ammonium to nitrate via nitrate, the process being carried out by the oxidation bacteria found in soil microbial population. The nitrate contamination can have negative effects for our health, and in this way, the development of a flexible, miniaturized, and rapid analytical device for this analyte determination was represented a necessity. We all know that in last decades, the nanomaterials have gained a high interest in biotechnology and bioanalytical development. And by using nanomaterials, it can be reached low detection limits, high selectivity and sensitivity in determinations. Our work involved the modification of the commercial screen printed electrodes with nanomaterials, nanocomposition materials obtained by the dispersion of multi-walled carbon nanotubes and metallic nanoparticles into different matrices of ionic liquids, sol gel, diaminonaphthalene, and chitosan polymeric films. To characterize and optimize the electrodes, the cyclic voltammetry studies were used, and for nitrate quantification, we have used the amperometric technique. For characterization of the electrode surfaces, the scanning electron microscopy and the Fourier transform infrared spectroscopy were used. So the scanning electron microscopy images show the coverage of the electrodes without and with surface modification. In the case of multi-walled carbon nanotubes, chitosan modified sensors, the roughness of the surface was increased, which was a sign of an enhancement of the electroactive surface area. On the other hand, by using silver nanoparticles, the nanoparticles showed an agglomeration of them into the nanotubes. And moreover, several cracks were observed in case of using sol gel as matrix. The Fourier transform infrared spectroscopy confirms the uniform formation of nanomaterial layer and the entrapment of the multi-walled carbon nanotubes and nanoparticles into the sol gel and polymeric films. In a first attempt, cyclic voltammograms were recorded by using sensors modified with different matrices. In the case of using diaminonaphthalene films, no signal was recorded. And even if the electrocatalytic behavior of the multi-wall carbon nanotubes sol gel based sensors seems to be the most improved, the peak wasn't well defined. By using silver nanoparticles, no enhancement in the signal was recorded, while by using multi-wall carbon nanotubes chitosan polymeric films, the peak was well defined through nitrate oxidation. So further, the study was based on using sensors modified with only multi-wall carbon nanotubes and chitosan in 1 millimolar nitrate in acetate buffer. And as it can be observed, the oxidation, the most increased oxidation peaks were obtained in the case of using nanocomposite modified sensors. For this reason, the multi-wall carbon nanotubes chitosan-based sensor was after used in the study of the analyte concentration influence. And as it can be seen, the increase of the peak potential intensity took place with the increase of the nitrate concentration ranging from 0.5 to 5 millimolar nitrate. Moreover, the stability had been proven by keeping the electrodes several weeks immersed in soil solution. In order to achieve a low detection limit and a good selectivity, the selection of the working potential or working applied potential had a really great importance. The kinetic nitrate oxidation at the surface of the electrode was studied with different scan rate values, and the oxidation peak intensity increased with the increase of the scan rate, and the linear dependence were obtained between the oxidation peak intensity and the square root of the scan rate, thus showing that the process is controlled by diffusion. On the other hand, the oxidation of nitrate to nitrate leads to an accumulation of nitrate on the surface of the sensors, and it leads to a passivation of the surface. The electrocatalytic behavior of the nanocomposite layer towards nitrate oxidation was studied at different pH values ranging from 5 to 9. And as it can be seen in this figure, with the increase of the pH values, the peak potential values were shifted to more positive values. The nanocomposite was highly electroactive towards nitrate oxidation in acid medium, while at neutral and alkaline pH, the activity decreased. Because we wanted to reuse the the sensors for several determination. We regenerated them in a simple manner by using cyclic voltammetry until the oxidation and the reduction peaks have decreased. For the nitrate detection, it was also carried out the amperometric study. And the analytical performances obtained at different pH values and different working applied potentials were obtained by using calibration curves with the linear regressions. And it was the results showed the fact that the nanocomposite based sensors showed good characteristic in both pH values at 5 and at 7 value 2. But especially in acid medium, the specific sensitivity has increased. The next step in our study was the detection of nitrate in real samples. In this way, different soils were collected from vegetal garden and from the cornfield. And these were used in laboratory for cultivating tomato seeds. The sensors were easily immersed in the low volume suction lysimeters, and by using this portable detector, the nitrate detection was easily performed from the soil solutions. The results obtained in real samples were compared with the classical spectrophotometric and colorimetric studies, and it was showed a good correlation between the results. And because we wanted to see if the sensors showed selectivity, an interference study was also carried out by using analytes known to be possible interference from the soil matrices. And the results show that no considerable influence was observed. As a conclusion, the flexible nanocomposite-based sensors were used to ensure selective, sensitive, and rapid determination of nitrate in soil by using a simple method. The developed sensors were characterized and optimized, and they were used in real samples detection. And for further, and as a perspective, we want to detect more analytes, because the sensors can be easily adapted for different types of potential contaminants in soil. I acknowledge the financial support from ManuNet NitroSense project, and I'm so grateful for Mrs. Doni for giving me the opportunity to be involved in this project during my internship at NeXT-Chem. I'm so thankful for my mentor and my colleague, and for our partner involved in the project. And I want also to say thank you to all my team. And thank you for your attention.
Thank you very much. The presentation was very good. If I understood well, this is your first presentation? Okay. Yes. I want to ask you if you used a commercial multi-well carbon nanotubes, or one with a pre-treatment.
The multi-well carbon nanotubes were commercial.
Okay. If there are any questions.
Yes,
If
Nikki
not,
Varaki speaking.
thank you very much.
Nikki, do you hear me? Hello? No?
So, please, there is a limit for the 10 minutes on each presentation, so be in time. The next speaker is Andrea Miron from abroad with the presentation entitled efficient copper removal from simulated wastewaters using alginate-based polymeric beads. If you are here.
I am here. Everyone hear me? It's okay?
Okay, please.
1 second. If you see my presentation, I think now it's okay, right?
Yes, and do your presentation on, okay, on slideshow.
So,
Thank you.
good afternoon, everyone. My name is Andrea Miron. I'm a researcher at Team 1, and I want to present a part of my results from Portugal and Romania, and for my intern researcher grant. So the presentation is structured in several chapters: introduction, aim, materials and method, results, and final with conclusion. Beads based on alginate and composite have attracted the attention due to their remarkable properties. They are used with different types of inorganic and organic fillers, such as carbon nanotubes, nanomaterials, and they are used in many fields, such as heavy metal absorption, dye removal, and antibiotic removal. The aim of this study is development a suitable procedure for obtaining beads for heavy metal absorption. The synthesis of the beads, it's easiest. The first step of the synthesis is the homogenization of the alginate in water and put the titania or the composite in the mixture. And the reaction conditions are 40 Celsius degree, 4 hour, and magnetic stirring. The next step is to put the warm solution in a syringe and drop it in a calcium chloride solution. And after 12 hours, the beads are washed and lyophilized. Here we see in the table the composition of the sample. One of the samples are with the titania obtained by a hydrothermal method, and the other one is based on a composite with titania and polyacrylonitrile. In the stir spectra, we can see all the characteristic peaks for alginate. We have the peak characteristic for the bonds COO, CH, and OH, characteristic for the alginate. And at a value of 2043, we have the characteristic peak for the alginate, for the cyan group in the polyacrylonitrile. XRD analysis reveal data about the crystallinity of the sample. We can see the peaks characteristic for anatase phases in titanium dioxide. And in the case of the composite, we can see the peak at a value of 17 degree, characteristic for the polyacrylonitrile. In the case of alginate, we can see amorph structure. The TGA results and DTG relieve data about the thermal stability of the sample. Of course, in the case of beads based on alginate and composite, we can see the characteristic degradation for the composite, for the polyacrylonitrile. And in the case of the beads based on alginate and titanium dioxide, we can see a higher stability because the titania, because it's an inorganic material, it's stable in this range of the temperature. Same images reveal data about the structure of the beads. in the case of beads based on with TO2, TO2, sorry, we have a uniform structure compared with the beads with composite, and the SIM images show this. For the absorption capacity, I make some preliminary tests. The first test it was based on solid-liquid ratio, and I used four different type of solid-liquid ratio: 0.5 milligram per milliliter, 1 milligram per milliliter, 2 and 1.5. And if you see here in the graphics, the best solid-liquid ratio, which was for 1.5 milligram per milliliter. The next step of the preliminary tests consists in choose the best pH for the absorption study. In this case, I used pH of 6. It's more or less the pH of distillate water, pH 5 and 4. And of course, the best pH when I choose to work in the future, it was pH 5, because they are the best absorption capacity. And in final, I make the kinetics absorption study. We can see in the case of sample with composite, the absorption capacity is higher compared with the beads based on alginate and titania. And the absorption capacity increased to more than 500 minutes, and after that they are stabilized. So in conclusion, alginate and composite and titania beads was successfully obtained. The beads present all the characteristic bands in the FTIR spectra. XRD spectrum suggests the presence of the characteristic titania peaks and the composite in the beads. From SEM images, we can see the uniformity of the beads, and the optimal condition for the absorption study, it was pH 5 and solid liquid ratio 1.5 milligram per milliliter. And the kinetic study relieved data about the comportament of the beads in solution with copper. So that means the beads with composite, it's high. The absorption of beads of composite is higher compared with beads with titania. Thank you for your attention.
Thank you, Andrea.
Thank
It
you.
is a very good work. So good luck in the future.
Thank you
I
very
want
much.
to ask you if you thought for using these materials for another metal, to remove this metal in the future from the wastewaters?
Yes, I tried to make some tests, but for now I have just preliminary tests. I start to make tests with nickel and a mixture of nickel and copper. But it's just some preliminary test. I need more time to work of this part.
Okay, thank you. If
Thank you.
are there
Thank you very much.
any questions in the room or online? If not, we can go to another speaker. The next presenter is Marinela Dumitru, also from abroad, with the presentation entitled Cryogel based on chitosan for antibiotic retention.
Hello, you can hear me?
Hello, good luck.
Thank you very much. Can you see my presentation? Okay, so I will start. My name is Marinela Victoria Dumitru, and I will present my study, cryogels based on chitosan for antibiotics retention. My presentation will contain a short introduction, the materials and methods, sample characterization, and finally some conclusions. In the introduction, I would like to talk about antibiotics, which are one of the most powerful drugs. They are used to treat several diseases, and unfortunately, due to their uses, they are becoming a real problem for water pollution. So in order to solve these issues, hybrid cryogels based on chitosan were developed. Cryogels are polymeric materials prepared by cryotropic gelification using polymers or monomers, hydrophilic or hydrophobic as precursor, being formed by stages of freezing and lyophilization. Cryogels are also used for several applications such as bacterial proliferation, drug delivery, biosensor cell delivery, and many others. In order to obtain the materials, two stages were needed. The kaolin silylation, because it's an inorganic compound, and when it's incorporated into the polymer matrix, need to have some organic groups because chitosan is an organic compound. After the incorporation of the clay, was added a physical and cross-linking foaming agent. And after that, the sample were freezed and lyophilized. For my first studies, I was trying to make some tests in order to obtain the best recipe in terms of the quantity between the chitosan and the solvent, and also the clay and the chitosan, in order to have a more resistant structure of the cryogels, and they can resist in time, much more time. From this slide, it may be observed that several tests were made. T2, it was the best one, and also from the swelling degree can be confirmed that sample T2 was able to swallow a lot of water, and also the resisting time of it was like 3 hours. From the FTIR spectra, it may be seen in figure 4 that the silicate, the modified silicate, was successfully incorporated into the polymer matrix of chitosan. Also, the specific bands for chitosan are can be seen from this spectra. Carbon, oxygen, carbon dioxide, oxygen, amine, carbon, oxygen, and many others, and also the silica, oxygen, aluminium. For this study, I was choosing carbamazepine and ciprofloxacin as the two antibiotics that I want to test my sample for absorption them. And in this slide, it can be seen in figure 5, the pH values for the antibiotics concentration. This concentration were actually used also for the calibration curves. And in here can be seen that the calibration curves were actually having a very good values in terms of air value. It was like 0.9999, a lot of 9. From the same images, it can be seen that the silicate was successfully incorporated into the structure, and also the samples show a spermatophore structure. That means they are able to absorb water and also antibiotic. I was trying to make for carbamazepine the friendly isotherm, and also from table 1 it can be seen the removal capacity of sample P6K for carbamazepine. It's like 59% of removal capacity for my sample, which is a very good behavior. For ciprofloxacin, I was able to do the both isotherms, and I was finding the equilibrium concentration absorption, actually, what I was going to use for the kinetics. Also for this, for table 2, it can be seen the removal capacity of my sample. For ciprofloxacin, it was like 56%, so it's a very good result. And I will going to do more of them. I was also making the kinetics, but I didn't have time to prepare and to make graphs for this presentation. To conclude, new hybrid cryogels based on chitosan containing modified silicate were developed with the FTIR spectra confer the incorporation into the inorganic clay into the polymer matrix. The swelling degree proved that all the cryogels are able to absorb large amounts of water. Same images cover the true macropore structure of the materials and the successful incorporation of the modified silicate. And from long wave and from the isotherms, it may be observed the absorption capacity for the samples, which is very good for both antibiotics. I want to mention that all these results are obtained at Coimbra University on the chemical department and the engineering department with the help of Professor Luisa. Her picture is in the left. And also I had the opportunity to work with some of their group, some persons from her group. And it was a very good experience. And also the results were actually okay. And I can do a lot more. Thank you very much for your attention.
So thank you, Marinela, for this interesting results. We have time for one or two short questions. No? I have one. Why did you choose those antibiotics?
Ciprofloxacin and carbamazepine are the both antibiotics are very used because carbamazepine is used to treat epilepsy and the ciprofloxacin for bacterial infections. And due to their high use in this medical field, they are actually existing in several amounts in the water, unfortunately.
Okay. Thank you. Good luck.
Thank you very
And
much.
see you on the next presentation. Next speaker is Andrea Luisa Mertz with the presentation entitled Bioactive Glass Particles for Bone Tissue Engineer. So please.
Hello, I am Luisa Mertz, researcher in Team 2 at NeXT-Chem. And today I will provide you with an introduction in my PhD topic, which is bioactive glass, or shortly bioglass, used in bone tissue engineering. I will talk about the theoretical part and also the experimental data that I obtained. Bioglass is a material discovered by Professor Larry Hench in 1970, and since then it has been really studied in biomaterial used as used in bone tissue engineering. The bioactivity of bioglass the bioactivity of bioglass is due to his ability to initiate a biological response in contact with body fluids. When it's implanted in the body, this bioglass forms hydroxyapatite layers, which mimics the natural bone composition. This hydroxyapatite layer can facilitate the attachment of cell bonds and leading to bone formation. It's a biocompatible material because it's well tolerated by the body, and it is a versatile material because it has a various range of composition and properties. If we talk about structure and composition, here we have a ternary diagram phase where we can see the relationship between this oxide composition, silicon dioxide, calcium oxide, and sodium oxide. So we can see the relationship between this composition, this oxide composition, and their ability to bond to hard or soft tissue. We have the structure of bioactive glass, and we can see we have network modifiers and the network former. In this case, the silica and phosphorus pentoxide works as network formers because they are able to create the glass skeleton. And the calcium oxide or sodium oxide are the network modifiers because they can add another oxygen. And in the table 1, we have the most common composition of bioactive glass, starting with 45S5 bioglass obtained by Professor Hench. We have 2 methods of synthesis. The first one, melt quench method, where our precursors are mixed and heated to a high temperature to obtain melt material. This material is then poured into a mold or in a liquid medium in order to obtain monolithic glass or glass powder. It is a simple process with it is cost effective, and we can obtain in a large scale this product. As disadvantages, we can mention the high processing temperature and limited composition control. The second method is the salt gel, where our precursors are mixed, and in order to have the hydrolysis and condensation reactions to form a sol. This sol is then subjected to aging and drying to obtain a gel. This gel is then heated to enhance densification. As advantages, the salt gel method is lower processing temperature, and we can obtain high purity materials. However, it is a time-consuming process and may require higher costs. Bioglass has various applications due to its unique biocompatibility. It is used in bone regeneration or as a carrier in drug delivery, in dental restoration and oral health care, and coating material for implants to improve biocompatibility. I've done some synthesis of a bioglass in order to obtain a 45S5 bioglass using these percentages and these oxides. In the first synthesis, I used a surfactant to improve the specific surface area. Both syntheses are similar. The gelation time is different. In the first synthesis I've done, the gelation time is 10 days, and in the second is just 24 hours. But in the end, I obtained a white powder in both syntheses. These materials were analyzed by scanning electronic microscopy, and we can see that in the first synthesis we have more porous structure. And also I obtain using the nitrogen absorption desorption a higher specific surface area. But we can see that both materials tend to have small particles that tend to attach to the larger ones. The thermal analysis, we can see that we have some mass loss. No, oops, mass loss here, where my material lose the water and some volatile compounds. We can see here some endothermic peak due to melting process, and then exothermic one due to crystallization process. In conclusion, we discovered the bioglass as a biocompatible and bioactive material by a sol gel technique, which can be used in various biomedical applications. To further investigate the relationship between the parameters, the process parameters and the concentration of precursors, we aim to do more experiments to obtain more exact data to say. Thank you for your attention.
Well, thank you, Andrea. If you have any questions. I have not this time. So the next speaker is Robert Alin Pele, discrete in, yes, Ionuț Octavian Zăuleț
I'm
is
present.
the next one with the presentation entitled Marbles versus Granite, weathering in different climatic environment. So please.
You hear me? You hear me now?
Yes.
Of course.
It's okay.
I'm for presentation. I refer marbles versus granite, whatever in different climatic environment. For the introduction, I referring the majority of the exterior stone structure and decoration are subject to temperature variation, both day and night. Alterations such as granular disintegration, contour scaling, and alveoli formation can be observed either at the base of the blocks of column or the median height. Aim of the paper, I've simulated the cycle of the temperature or light of sample exposed to 25 cycles. Each cycle consisted in 6 hours of exposed to 45 degrees and 76 humidity, after which the sample were exposed to room temperature and cooled down. The materials I've used is 3 types of the marble: Ruskisa marble, Albesh marble, and Carrara marble. 3 types of granite that come from Italy: Rosa granite, gray granite, and base granite. Equipment where I use is chromatic parametric using a Konica Minolta and climate chamber Smart Pro. Chromatic parametric, the result is the before and after exposure temperature is decreased after 25 and 25 cycles, and the same is the just a moment the marble, much less for granite. In the conclusion, the treatment in the climatic chamber causes mechanical disintegration of the outer part of the marble and granite, starting from the discontinuity contained in the rock and between the face, the different mineral that from the stone. Color change can indicate the damage degree. The chromatic parameter indicates and increase the luminosity during the thermal temperature and climate chamber. A slightly decrease of the parameter red grad and decrease granite or increase marble of B parameter yellow blue more pronounced of marble versus granite. Marble is more sustainably at the external factor than granite. And thank you for your attention.
Okay, so thank you, Mr. Zaulets. I have a small recommendation for the futures presentation. We want to see you on the camera.
Sorry.
Also, we think you are a ghost. So
No, I know. I am
okay,
human.
if you have any questions. If not, the next speaker is Roberta Lynn Pele with the presentation entitled Discrete and Extending Structures based on cobalt and hexafluoroacetylacetone.
Hello, can you hear me?
Hello.
You can see the presentation?
Yes.
Okay.
All good.
Hello, my name is Roberta Lin Pele. I'm a student at the Faculty of Chemistry, University of Bucharest. And today I will present my work regarding discrete and extended structures based on cobalt and hexafluoroacetylacetone. As an introduction, to motivate my choice of study, we know that coordination complexes of transition metals and also of cobalt have many interesting applications in optics, catalysis, electricity, magnetism, especially with a focus on single molecule magnets, and many other interesting applications. So we try to obtain such compounds starting from cobalt, which is a versatile metal ion. It has multiple possible coordination geometries and it can display magnetic properties. And for our ligands, we had some carboxylate ligands. We used diethanolamine, but most importantly, we used hexafluoroacetylacetone. This organic ligand has a high density of electronegative fluorine atoms, which can lead to intermolecular fluorine-fluorine interactions, which have been shown to enhance the crystal structure and also the magnetic properties. The fluorine atoms can draw the electronic density away from the metal, which would also lead to better coordination. Starting from this, after many trials, we obtained 7 new coordination compounds. The first one, here we have the synthesis. We started from the diethanolamine, which was deprotonated by 3-ethylamine. Then we added cobalt hexafluoroacetylacetone salt. I will abbreviate hexafluoroacetylacetone as HFAC, if you don't mind going forward, because it's a very long word. And then we obtained these dark green crystals that you see on the slide after the evaporation of the solvent. We characterize them by single crystal X-ray diffraction. And here we can see the structural unit of the compound on the left, with 2 anionic hexafluoroacetylacetone molecules as lattice anions. The molecular unit has a hexagonal disc-like topology with 7 cobalt ions, out of which we have a central cobalt-2 ion. And on the outside, we have alternating cobalt-2 and cobalt-3. We can see that the hexafluoroacetylacetone is connected as a capping ligand on the cobalt-2 ions and the diethanol between the various cobalt ions. Each metal ion has an octahedral geometry, as we can see here on the right. We performed the spectroscopy studies, Fourier transform infrared, to confirm the presence of the organic ligands. Going forward, we have a compound, actually 2 compounds mixed because it's the same structure, but we have different lattice anions. One has a perchlorate and one has the tetrafluoroborate. The synthesis is very similar, but here we used 2 salts. We used cobalt HFAC and cobalt perchlorate and cobalt tetrafluoroborate. Again, we can see beautiful dark green crystals, which were characterized by single crystal X-ray diffraction. And the structure is very similar. Actually, the molecular unit is identical to the first compound, but we have different crystal lattice anions. We have perchlorate and tetrafluoroborate, whereas for the first one we had hexafluoroacetyl acetone. To confirm that we have the lattice anions, we performed the infrared studies, and as we can see here, there is a difference in the signal around 1,000 centimeters to -1, which confirms the presence of the perchlorate and the tetrafluoroborate in these two compounds. For all first three complexes, we have here the UV spectrum to confirm the presence of the metal ions in their respective geometries and oxidation states. And as we can see, they are very similar because we have the same metal ions in the same geometries, in the same oxidation states. Going forward, we have a heterometallic polynuclear compound with cobalt and copper. We used again the ethanolamine, and then we added copper H5 salt and the tetrafluoroborate cobalt salt. These crystals were again characterized by single crystal X-ray diffraction. And as we can see here on this slide, we have a molecular unit accompanied by two tetrafluoroborate crystal lattice anions. And again, the hexagonal disc-like topology. In this case, we have a central cobalt 2 ion. And on the outside, we have 4 copper 2 ions and 2 copper 3 ions. The H5 molecules connect to 2 copper ions. And also the other left copper ions have a diethanolamine molecule connected to them. The cobalt 3 atoms are the same like in the last compounds. They have diethanolamine bridging ligands. Again, hexagonal octahedral geometry of the metal ions. The same studies for the organic ligands. We can see also the boron-fluorine bond here. And in this case, in the UV spectrum, we have specific bands for copper 2. So it's the same spectrum, but we have an additional band at around 850 nanometers for copper. The next compound is a tetranuclear compound, which was obtained starting from cobalt H5 salt, adding acetic acid, which was deprotonated with 3-ethylamine. We have here the solvent, which is going to be important going forward. In this case, we use hexane, methanol, and chloroform, and we obtain these pink crystals. As we can see here, they have a square-like topology with 4 cobalt 2 ions, each having 2 H5 molecules as capping ligands. They are in the cis conformation of the octahedral geometry, and adjacent cobalt ions have delta and lambda conformations. Here we have the crystallographic parameters. We have a crystal lattice 3-ethylamine molecule. And on this slide, I wanted to show you the crystal packing of the compound. We have this columnar packing of tetranuclear units. And on the right, we observed that we have the fluorine-fluorine interactions that we are looking for. We have a distance of 3.12 Armstrong between fluorine atoms from 2 different molecules. Again, the infrared spectrum to confirm the organic ligands. The next compound has the same synthesis as the tetranuclear compound, except we used a different solvent. We used absolute ethanol, and we obtain a coordination polymer. As we can see here, we have units of a cobalt 2 ion with 2 H5 molecules in the transposition in this case. So for the first one, had a cis conformation and bridging acetate ligands. We also have lattice anion as 3-ethylamine. On the right, we can see 2 chains with the octahedral geometries of the cobalt ions and the crystallographic parameters. Again, we notice fluorine-fluorine interactions as we have short distances between fluorine atoms from different chains, 3.29, 3.28 Armstrong, and also some hydrogen bonds that strengthen the crystal structure between the hydrogen from the triethylamine and the oxygen from the acetate ligands. In the UV spectrum, we notice the difference caused by the different coordination of the hashfaq molecules. So for the tetranuclear, I said that we had the cis conformation. For the coordination polymer, we have a trans conformation, and this results in a slightly different splitting of this adsorption band at around 400 nanometers, and a slight shift of the band here at around 500. The next compounds were mononuclear. We had a mononuclear cobalt hashfaq with acetonitrile molecules, starting from a hashfaq salt redissolved actually in acetonitrile. And this is the crystal structure. We have a mononuclear structure of cobalt with two hashfaq molecules in the trans positions, and the other two positions occupied by acetonitrile molecules. Here is the crystal packing of this compound. And again, we suspect that we have fluorine-fluorine interactions at 3.25 angstrom between fluorine atoms from different molecules. These are the spectroscopy studies. In the infrared, we notice a band for the carbon-nitrogen triple bond to confirm the acetonitrile. And the last compound that I will present, we have again redissolved cobalt hashfaq salt, this time in chloroform with triethylamine. We noticed that we obtain a different structure from the starting salt, which only had two hashfaq molecules connected to cobalt. In this case, we have three hashfaq molecules connected to cobalt, as we can see here on the left. And as a lattice anion, we have triethylamine molecule. On the right, we see intermolecular interactions. We have again fluorine-fluorine possible interactions at 3.23 angstrom and hydrogen bonding between atoms from the hashfaq molecules. As some conclusions and perspective, we successfully synthesized 7 new coordination compounds of cobalt with the hexafluoroacetylacetone ligand, as well as with other ligand molecules. They were characterized by single crystal X-ray diffraction, Fourier transform, infrared, and UVB spectroscopy. Going forward, we want to study the possible applications of these compounds in fields such as magnetism, catalysis, or optics, but especially magnetic properties, because we suspect that if we have these fluorine-fluorine interactions and these cobalt oxidation states, their single molecule magnet behavior. And we could also make a parallel between the mononuclear complexes and the polynuclear ones, because in the case of polynuclear compounds, there could be special spin couplings with more metal ions in one molecule. That's it. Thank you for your attention. If you have any questions, I would be happy to answer.
Thank you once again, Mr. Pele. If you have any questions from online or in a room. If not, thank you once again. So the next speaker will be Andrea Luca with the presentation entitled Oxide of Vanadium Complexes with Potential Insulinomimetic Activity.
Yes, hello.
Hello, also from online. So please present your result.
1 minute. Just 1 second. I have a little problem. So I think you see my presentation now. So, my name is Andrea Luca, and I'm here to discuss with you about some oxidovanadium complexes with potential insulin mimetic activity. So today I will talk about the toxicity of vanadium and its compounds, some form of vanadium in different body cavities and tissues, the mechanism of action of vanadium compounds with insulin mimetic activity, the synthesis way we obtained our compounds, how we characterize them structural and spectral, and the way we determine their anti-diabetic effect. So we know that vanadium is used as an active center in enzymes by various lime forms, such as vanadium bromoperoxidase in ocean algae and nitrogenesis in bacteria. Because vanadate structure and biology is similar to the one of phosphate, it can be viewed as a phosphate competitor in number of biological enzymes such as kinases or phosphatases. We know that the accumulation of vanadium can be fatal, or sometimes it can lead to carcinogenesis, neurotoxicity, gastrointestinal complications, and many other health problems. But we hope that our compounds are not toxic, but on contrary, they have a biological effect that we can use in our benefit. So we take vanadium primarily by two species as vanadates and vanadils. And we know that vanadium 5 molecules are largely reduced in the stomach before precipitating in the alkaline intestinal media to create sparingly soluble vanadium oxyhydroxide. But the vanadates are easily absorbed in the gastrointestinal system and is absorbed 3 to 5 times more effectively than the vanadil. Of course, different vanadium species are preferred depending on the solvent area, and we know that each of the species can form various complexes and has different chemical characteristics, which led to unique biological reaction in fluids, tissues, or even cells. But it's very important to understand their chemical bioprocessing to comprehend their mechanism of action. Here are some compounds that we know that have insulin mimetic properties, such as bis-maltolate oxidovanadium or bis-ethyl maltolate oxidovanadium. The interesting fact is that in this moment we don't know exactly what is the mechanism of action of the vanadium compounds with insulin mimetic activity, but there are 3 putative mechanisms which they can act as insulin, as stimulation of insulin receptors, effects on protein tyrosine phosphatase, or inhibition of secondary messengers. We tried to synthesize an oxidovanadium family, and we started from salicyl aldehyde or orthovanillin with an amino acid. In this case, the amino acid is valine. And we obtained this ligand, and the structures we obtained when we added vanadyl sulfate trihydrate. It is one presented here, which is a mononuclear compound, and here is a dinuclear compound. You can see here the crystal structures of our compounds, on which we made our biological studies. You can see here they are both dimers and they are dinuclear. Because we wanted to characterize our compounds before seeing their biological properties, we present you here the electronic spectra of oxidovanadium complexes in the left image, and in the right one you can see the electronic spectra of their precursors such as valine, salicyl aldehyde, orthovanillin, and vanadyl sulfate trihydrated. We can see the in the absorption spectra of our 4 oxidovanadium complexes that they display 3 main bands in phosphate buffer solution. We also made a study with circular diffraction, and we see that they confirm the findings of our X-ray crystallographic data. Of course, we wanted to study the stability of our 4 oxidovanadium complexes in phosphate buffer solution at physiological pH, which is 7.4, and the physiological temperature, which is 37 degrees Celsius. And we monitored this stability with UV-Vis spectrophotometry. And we see that our compounds are decomposed in a time-dependent manner when they are incubated in PBS. And this fact is indicated in the decrease of the intensity in a specific wavelength. We used fluorimetry assay to monitorize the capacity of the oxidovanadium compounds to bind to the bovine serum albumin. And the result was very interesting because we saw that our compounds can possesses albumin binding properties up to 3 hours. So that's why we performed an extensive study to determine the biological activity of our compounds. We studied the effect of our compounds on alpha amylase activity, on hepatoblastoma cell line, cell viability, on total protein tyrosine phosphatases, and on insulin receptors phosphorylation. And we saw the fact that our compounds have an amazing insulin mimetic activity, and they do not show cytotoxic effects on hepatoblastoma cells, and they also have an inhibitory concentration 50 higher than the observed for cisplatin. At this moment, we try to obtain a new family of oxidovanadium 5 complexes with Schiff-based ligands, but this time they are derived from orthovinylin and isoleucine and leucine. The interesting fact that we observed is the compound which has the isoleucine precursor is a mononuclear dimer, and the one which has the DL isoleucine precursor is a dinuclear compound. Here are the IR spectra because we wanted to confirm our structures. And here are some videos, I hope you can see them, to see that one of the compounds is dinuclear and they both have the S and the R enantiomers. And here is the mononuclear compound, which also have the S and R enantiomers. In these structures, our vanadium is in 5 state, and his geometry is a disordered octahedral. In conclusion, we developed some oxidovanadium complexes, and they were characterized spectrally and structurally, and all our complexes exhibit stability in solution at physiological pH and at 37 degrees Celsius for up to 3 hours, and have a moderate to high rate of decomposition after this period. And this is the thing that they suggest the fact that they decomposed in biological media. We also saw the fact that in vitro they have anti-diabetic properties and insulin mimetic effects in all the 3 mechanisms of action, which is the reduction of alpha amylase and the total phosphotyrosine activity, and they increase the phosphorylation of insulin receptor in hepatoblastoma cell line. And we hope that our synthesized oxidovanadium complexes may be a very good candidates for further in vitro and in vivo biological analysis. And we hope that sooner or later they will be used as treatment for patients with type 2 diabetes. Thank you for your attention. And if you have questions, I am gladly to ask them.
So thank you very much, Ms. Luca. Was a good presentation with good results and good future perspectives. So if you have any questions for her, if not, thank you once again and
Thank you so much.
So the next speaker will be Andrea Miron once again, with a presentation entitled Synthesis and Evaluation of Novel Chitosan-Based Materials for Efficient Removal of Heavy Metal Ions from Wastewaters. Hello. Please.
Sorry. I think I don't have the control of the presentation. Oh, here it's okay. Thank you. So the presentation of today, it's synthesis and evaluation of novel chitosan-based material for efficient removal of heavy metal ion from wastewaters. The presentation is structured in introduction, aim, materials and method, results and conclusion. Chitin is the second one most abundant biopolymer in nature after the cellulose. We found chitin in an exoskeleton of arthropods and some marine sources like crabs, shrimps, and other one. And one of the properties of this material is insolubility in water and in an organic solvent. From chitin, by the deacetylation process, we obtain chitosan, that is a semi-crystalline solid. It's one of the most used natural polymers in many fields, such as pharmaceutical, cosmetic, water treatment, and other field area. The aim of this study is synthesis of chitosan from shrimp shell waste and obtaining the advanced polymeric material with the ability to retain heavy metal from wastewater. The chitosan synthesis is easiest. We start with shrimp shell waste. They are washed and grinding. After that results in a powder. The powder are lyophilization and freezing and lyophilization. And the next step to obtain chitin is the deproteinization of the shell. After the deproteinization, with a low concentration of natrium hydroxide solution for 24 hours at 25 Celsius degree. The next step is washed, freezing and lyophilization. After this, we have chitin with amount of calcium carbonate, because the normal process of obtaining chitosan involves two steps, deproteinization and demineralization, but we are making just the deproteinization of the shell. And the next step is the acetylation with a strong natrium hydroxide solution, 50% at 80 Celsius degree for 6 hours. After the acetylation process, the powder are washed and drying by in the oven, and in final obtain calcium carbonate, chitosan enriched with calcium carbonate. Here in the table we have the composition of the sample. The synthesis of the beads, it's easiest and it's a fast process. The first step is dissolving chitosan in acetic acid solution 10% at 60 Celsius degree 4 hour and the magnetic stirring. After that, the solution are put in a syringe and dropping in a natrium hydroxide 4% solution. After some 12 hours, the beads are washed with distillate water on the neutral pH and dried by lyophilization. So here we have the FTIR spectroscopy for all the sample. We can see here all the characteristic peaks for important for the chitosan and chitin. The most important bands in chitin are for the amine 2 group at 1550 and 1370. And in X-ray diffraction, we have the characteristic peak. And in the case of chitosan from the shrimp shell, we can see here at 29 and 40 degree, the characteristic peaks for the calcium carbonate. TGA analysis provide information about the thermal stability. We can see here we have the higher value for the chitosan made by the shrimp shell compared with the commercial chitosan and the chitosan obtained from commercial chitin. We have a value of 305.40 Celsius degree compared with the other type of chitosan. We can see in the case of chitin, we have the higher value of the temperature in the range of the TGA analysis. In the case of porosimetry of the sample, we can see that the chitosan obtained from shrimp shell have the higher bed surface area, and the pore surface area is higher compared with other type of chitosan. Compared with, for example, compared with the commercial chitosan, have a pore surface area of 0.89. Compared with the chitosan obtained from shrimp shell, we have a value of 11.16. Same images show the presence of the calcium carbonate in the structure of the chitosan. You can see in image C the presence of calcium carbonate compared with the commercial chitosan and the chitosan obtained from shrimp from commercial chitin. The next step, which was to obtain some preliminary tests for the absorption capacity. I have to sample when I measure the chitosan obtained from shrimp shell and the beads with the chitosan obtained from shrimp shell and the beads based on chitosan obtained from commercial chitin. And I make the solid liquid ratio. So in all the cases, in both cases, we have the best solid liquid ratio, best for the ratio of 1.5 milligram per milliliter. So we have a capacity of absorption more than 60%. And in the case of this sample, the capacity of the absorption is higher for chitosan obtained from shrimp shell compared with the chitosan obtained from the commercial chitin. So in conclusion, chitosan from shrimp shell was successfully achieved. FTIR spectra show all the characteristic group for the chitin and the chitosan. The XRD spectra confirm the presence of calcium carbonate in the chitosan made by shrimp shell waste. The thermal analysis showed the calcium carbonate enrich the thermal stability of the chitosan obtained by shrimp shell compared with the commercial chitosan. Same images confirm also the presence of calcium carbonate in the chitosan, and we have a higher surface area for this type of chitosan compared with commercial one. And the preliminary tests for the absorption capacity show the optimum solid liquid ratio at 1.5 milligram per milliliter. Thank you for your attention.
So thank you very much,
Thank you.
Miss Miron. These are outstanding results. You proceed with various ideas.
Yes.
These are preliminary tests, but I think that it will be interesting if you discover other parameters for absorption capacities in order to evaluate the removal capacity from wastewater. I mean, you can use different pHs or different concentrations of materials or heavy metals.
Yes.
Future perspectives.
Yes. Thank you for the suggestion. I will make this test in the future. Thank you very much.
Okay, thank you once again. So I will go to the next speaker, Miss Marinela Dumitru, with the presentation entitled Composite Materials Based on Chitosan for Slow Release of NPK Fertilizer in Agriculture. If you are ready, Andrea.
Yes, you will start. Okay, so now I can give share to my. So hello again.
Hello.
I will present another part of my work. My work with Alin Vitila. We are doing this for the internal grant. I will present composite materials based on chitosan for slow release of NPK fertilizer in agriculture. Just a second. Okay. So I will have a short introduction, the materials and methods, sample characterization, conclusions, and some future perspectives. I will want to talk about the chemical fertilizer that are used a lot in our days. Unfortunately, they affect the environment. They also deteriorate the soil. They affect the water contamination and many others. In comparison with organic fertilizer, which actually enrich the soil with nutrients, the preparation of them involves a low cost and the soil quality it's improved. For our samples, we needed some steps. So the first step was obtaining a liquid digestate. It was actually made by my colleague Alin Vintila. He was using some organic waste, and through anaerobic digestion, he obtained the liquid digestate. After that, I incorporated the liquid digestate in my cryogels, and maybe seen in this slide the sample that I obtained. And I would like to have these materials for agriculture application to controlled release this type of fertilizers. From figure 3, it may be seen the swelling degrees for the cryogels. I was trying to make some tests in order to obtain the best recipe for the cryogel, and after that to incorporate. So I established, and I was choosing C4 because it was showing the best behavior in terms of the swelling degrees and also the resistance in time. After that, I was trying to incorporate different amounts of liquid digestate, and it can be also seen from this slide that the sample C4 end, it was showing the best results in terms of the swelling degree and also the resistance in time of the samples. From the FTIR spectra, it may be seen that the liquidity state was successfully incorporated into the polymer matrix by the appearance of a peak around 2150. From SEM images, it can be seen that the liquidity state was successfully incorporated into the polymer matrix, and also the materials show to have a macroporous structure which make them suitable for controlled release of fertilizers. To conclude, the new composite materials based on chitosan with NPK content were developed. The FTIR spectra successfully confirmed the incorporation of the organic waste into the polymer matrix. The swelling degree proved that all the cryogels are able to absorb large amounts of water. And from some images, it may be seen a macroporous structure of the composite materials. As future perspectives, we would like to test these cryogels and to determine their controlled release capacity for NPK. Thank you very much for your attention.
Thank you also. I have a question. Since we intend to use these materials for application in agriculture, I would like to ask you if you know what types of plants they could be used for this application.
We would like to try to test them on corn, on oat, this type of plants, because they are really need these nutrients, and the soil should be much enhanced with this quantity of NPK.
Okay, thank you for your answer. If there are no more questions. Okay, thank you once again. See you in the next minutes.
See you. Bye.
In the next few minutes, I will invite our young researcher went abroad for give us their impressions about exploring research internship from Portugal and from France. Toulon, exactly. So the next speaker is Julia Elena Neblea.
Yes, hello, can you hear me?
Yes.
Okay, I have a short presentation. I have a short presentation right away. Okay. Just a second. Okay. So I will say a few words about me, because I don't know if everybody knows the domain that I do my PhD right now, my study field. I'm a scientific researcher, a member of Team 1 in Advanced Polymeric Materials and Polymer Recycling Group. My study field for my PhD is in chemical engineering, about micro and nano gels based on biocompatible polymers. I study at University Polytechnica of Bucharest under the guidance of Professor Mircea Teodorescu. About the mobility exchange program, I am currently still here at University of Toulon in France. For this exchange program, I was here in France since 1st of March, and I am leaving on 31st May, so next week. The research field that I explored here was about nanogels based on biocompatible polymers under the guidance of Professor François Xavier Perrin in Laboratoire MAPIM here at University de Toulon. The main areas of research of this laboratory is the study of material degradation, developing and improving the performance of new materials by developing nanostructured, nanocomposite products, and by modifying their surface, the material surface. The main directions are with applications in biofilms and anti-fouling, and durability and functional materials. A few things about the activities I did here. So first of all, I came here in order to optimize my nanogel synthesis by inverse mini emulsion polymerization, the one that I talked earlier in my presentation. Also, I did some characterization techniques. I work with proton NMR, I work with infrared, with dynamic light scattering, with scanning electron microscopy, and also with an environmental scanning electron microscopy. I will say a few words about it in the next slides. And also what was super interesting for me, I did also the protein quantification, because before coming here, I did not work so much with proteins. I did not know exactly this biochemical part. So I learned new techniques like SDS page, electrophoresis, Bradford method, and different binding studies. So for these kind of activities that I did here, I had also financial support from Erasmus scholarship, which covered my accommodation and day-to-day expenses. And also the internal mobility grant from NeXT-Chem for young researchers. This covered my transportation to and from France. The challenges that and opportunities that I experienced here was firstly, of course, as I said, studied and optimized this polymerization process, inverse mini emulsion. I learned new techniques for protein quantification, as I said earlier, SDS page electrophoresis, Bradford methods. I studied the morphology of the nanoparticles in the environmental SEM. The environmental SEM is actually has the same principle as the normal SEM, just that in this kind of SEM, I studied the samples being in a solution. So the nanoparticles were dispersed in a solution. And even though they were partially dried, it allowed me to see them if they are swollen or not, if they take water, if they intake water or not. So the vacuum used in the environmental SEM is lower than the normal one. The samples did not need coating, gold or carbon coating, so it was really easy to work with. I was also trained to work alone with other different characterization equipment. I worked in a really interactive and multicultural environment. I improved actually my problem solving abilities because I had to share the laboratory with other people, which also were not native French speakers or native English speakers. So it was pretty interesting to learn about their culture, but also to see their techniques in the laboratory. So I had new things to learn from each of the persons that I met. I improved also my teamwork abilities, and I also improved my English and French communication skills because besides working with the Vietnamese colleague, Lynn, she was a Vietnamese and we talked in English, but I also worked with Tunisian girl and the Moroccan girl, which they are native French speakers. So they helped me a little bit with my French. My basic French. Now I can proudly say that I can have a basic conversation in French. Okay, this is just a normal day in the lab. A little messy, I know, but is chemistry ever clean in laboratory? In the lower part of the slide, you can see my workplace. Well, one of the days where my workplace is. And in the upper part is actually, this is a part from my colleague Lorena. She will present you the next presentation about her work here at MAPM, but it was pretty funny because I went there to say hi and ask her how she's doing and if she works a lot and things like that. And I saw everything written on the fume hood glass. And it was pretty funny because I also do that in my laboratory. It's easier to have the information exposed like in a whiteboard, you know, and when you're working in the fume hood, you just look up and see, oh, okay, that's what I need to do. And you just remember, okay, let's do that, let's do that. So it was pretty funny. for me, but it's also very practical. Okay, and some results so far. As I said before, the SDS page electrophoresis was pretty interesting to work with. I did not experience this kind of protein quantification before, so I did some gels, some trials on my proteins. I also did the microplate assay for protein quantification. This is the Bradford method, the applied Bradford method. The environmental SEM, as I said earlier, this in the right corner, right upper corner is the environmental SEM, and the normal SEM in the lower corner. These are the result, a few of the results so far. I know they are messy on this slide, but there are a lot of results that I still need to work on. I did not manage to finish everything in the lab. Actually, we're still working, even though next week we're coming back to Bucharest, but we're still working hard on these results. So the first picture here in the left corner is an SEM image, the normal SEM image, where we can see the size of the particles, so below 200 nanometers. And the other image is the environmental SEM image. Even though it's easier to work with environmental SEM because we can study samples in solutions, it's difficult to magnify more like in the normal SEM and to get a very good resolution like we do in the coated dried samples in SEM. I also have some DLS measurements, which are thrown away somewhere here in the slide, and also some UV absorbances for the proteins that I analyzed. Mainly, this is the work that I've done. There are a lot of results, but it was very difficult to put them all in just one short presentation. I'm grateful to my team in Romania for supporting me, calling me, answering me at every email and every telephone that I needed. I'm grateful to my mentor, Ana Maria Zaharia. I'm grateful to my colleagues, Mihaela, Vera, and Danita. I am also grateful to my colleague Lorena that she was here with me, and we actually supported each other in our work also in the lab and outside the lab. And I'm also grateful to my colleagues Andrea, Mari, and Sorin, that even though they were far away from us, we also communicated very nicely, and it was pretty nice to see that I have their support. So thank you so much for your attention. Can you hear me?
Yes. Thank you very much. It seems that you like this stage very much. So we are also lucky to see what results you have until now. So
yes,
thank you once again, and hope to see you soon.
Yeah, see you soon.
So in the next few minutes, I will invite Mrs. Ana Lorena Neagu to present her impression from Toulon.
Okay, just a sec, please. Can you see the presentation? It's okay?
Yes.
Okay, so hello again, everyone. So I'm gonna show and talk to you about my experience since I'm here at the University of Toulon. So this all internship for me takes place here at the University of Toulon.
Open your camera.
It's open.
Lorena.
It's open.
Okay. Okay, it's open. Okay.
Hello. Okay. So this internship takes place here at the University of Toulon in the MAPIAM laboratory under the supervision of the Professor Hugues Brisset. And it will end, unfortunately, on the 31 May this month. So it's a little bit sad, but it is what it is. So we actually went from we start from applying for a scholarship from Erasmus. We saw that the Erasmus scholarship also just covered the accommodation and the day-by-day expenses. So we also applied for the mobility grant, which from this way, I would like to thank you for the for this opportunity to arrive here. What can I say more than already Julia told? The first contact with the University of Toulon was mind-blowing. Everyone was quite nice. Everyone was so pleased to help you, to teach you everything. Was quite nice experience. I totally recommend for everyone to do so. So I would like to talk something about my professor here, Hugues Brisset. Is a professor from University of Toulon. Also is the head of the MAPIAM laboratory and has a lot of experience in the development of new material based on the electroactive molecular imprinted polymers and for sensor application. Also is a man with a lot of patience. He tries to explain to you so much times to make you understand what you're doing there and put so much patience in what he's doing. It's a total fantastic man. So for this internship, the proposed work plan was the synthesis of polymer layers by electropolymerization with embedded molecular imprinted polymer particles, also physico-chemical characterization of different polymer layers and the resulted sensors, and the electrochemical measurements of the obtained sensors. So we thought for first to try with ethylene dioxythiophene as electropolymerization for the electropolymerization as monomer. So for this type of application, in order to embed the molecular imprinted particles inside the layer and this layer to be on the SPCE, you cannot do this exactly from the beginning on the SPCE. You first need to establish some parameters, to do some tests, and then work on the SPCE. So this is what we try to do to make the electropolymerization on a 3-electrode electrochemical cell. So we had as the working electrode the gold electrode, the reference one calomel electrode, and the counter electrode platinum wire. And we use the cyclic voltammetry in order to do the actual electropolymerization. So first, you can see in this picture the electrochemical cell with the electrodes and everything, how the E-dot electropolymerization takes place to say cyclic voltammetry, the P-dot response, which was at first a total success. And this is the P-dot, how it looks on the gold electrode on the surface. So it was quite good. And then the struggle begins. So we realized we cannot do some analysis on the surface of the gold electrode because it's a long electrode. We cannot do another tests on them. So what we do next, we find the solution of making the electropolymerization on indium tin oxide glass. So at first we try to do the same technique with the same parameters, but the electropolymerization took place on the counter electrode instead of the working electrode, which is quite, and we don't know why. We're still trying to figure it out. So we said, okay, if this is not working, let's try something else. Let's change the counter electrode with another litho glass. Let's see if it works like this. So apparently it didn't. We tried to do the same thing, but the electropolymerization took place also on the working, but not such as good, but also on the counter electrode, and it's not okay. So after many attempts, we stopped and said, okay, let's change the method. So we changed from the cyclic voltammetry to colorimetric method. So it was a success till now. So first thing in an internship, be persistent. It will work in the end. So I just want to show you what Julia showed earlier, a usual day in the lab for us with all the sketches and notes and everything, just to make sure that everything is fine, everything went well, the synthesis went good and all. Also, here we had the occasion to learn how to use some apparatus. For me, I used the AFM and also the SEM apparatus. I learned how to use them because until now I didn't actually do some analysis on this type of apparatus. So for me, it was something quite new. So the second advice on an internship, never fear to learn something new. So it was quite useful for me, very useful also for the future. So these are also some results what I obtained till now. You also saw them in my previous presentation. And a short brief review of this internship till now. We learned new techniques, we saw new places, and we made such many friends here actually. So thank you all. I would like to thank also the colleagues from MAPM and also for Team 1 for their support and all for what you're doing. Thank you.
Thank you, Lorena. Very nice presentation. And
Thank
now
you.
we are passing in Coimbra, in Portugal, to Marilena Dumitru.
Just a minute. Yes, just a minute, please.
Please, Mari, 5
yeah.
minutes.
Yeah,
Yes?
yeah. I think I can share. No, I can't share the screen. Lorena should need to.
Just a second.
Okay.
Okay, we hear you. Okay. Put your presentation.
I cannot do it. I wait to Lorena to stop it.
Just a second.
Yeah, no worry. Okay.
Yeah.
Now I can share it. So, just a second. Okay, can you see my presentation?
Yes, it's okay.
Okay. So I'm very glad for the girls from IPN because they seems like they are having a very good time in there. I also have a very good time in here. It's a very good opportunity, and I will talk about my experience in here. My internship was actually supervised by Professor Luisa Maria Reyes. She is a very specialist person in aerogels, in pollutants removal from wastewaters, and many other applications. In this place, I was having the opportunity to see a lot and to learn a lot. University Coimbra is one of the most oldest one in Europe. It's was starting in 1219. It's a very big university with a lot of students, and they are having also enough equipment. They are developed on this part. They are having different direct contracts with private companies, and they help a lot of students. They form the students, and in here I was able to make some tests, to study my sample, to obtain some results that it could be helping me to finish my PhD thesis. it's a very good opportunity and a very good experience. I will not be able to come in here without the help of my team, of Tanța Verna Iordache and Mr. Andrei Sârbu. I'm very I don't have the words because it's a very good opportunity and it's a very good experience. I was able to work with different equipments and I was learning a lot. The girls for the department, they were master students, PhD students, every one of them, it was for a different part of the world. So we have French, we have Brazilians person, we have also from Portuguese, we have Portuguese, and they were helping me a lot and I helped them. We were just working in the lab and my English actually, it's much better now. From Professor Luisa, I can't have the words. She's a very good professor. She was the best supervisor because she was trying to help me, to talk with me, to advise how we are going to do, what we should do, and what we should make. So I really want to thank my team and also for the mobility grant, to Mr. Radu Cherescu, for all my colleagues and for all my supervisors. Thank you very much.
Thank you, Marie. Thank you very much too. We will expect you here in Bucharest.
Yeah.
And now, Andrea Miron. She will tell us her impression.
I am here. Hello again. I want to present my experience at the University of Coimbra from Portugal. The internship supervisor was Professor Dr. Artur Valente, and so I start with some short introduction about me. I'm a PhD student. I'm a research scientist in NeXT-Chem Bucharest in Team 1, Advanced Polymeric Material and Polymer Recycling. My interest domain are inorganic organic composite, advanced polymeric material for wastewater treatment, polymeric composite membrane, and polymeric composite beads. I come here to make a part of my PhD thesis and in the field of polymeric composite beads. And I find here a lot of nice people, a lot of colleagues. They are start to help me a lot. So the Coimbra University, it's one of the oldest university in Europe. Of course, my colleague Mary tell this, and they are a lot of faculty. The chemistry department, when I'm here, it's a part of the Faculty of Technology and Science, and the research group led by Professor Valente has a lot of has 3 PhD students, 3 boys. They are PhD students, and 5 master students. My colleague Eva is the lady with curly hair. He helped me a lot to make all my sample, to dilute all my sample, because usually a table from atomic absorption for heavy metal absorption look like this. In the first week, I start with 5 bottles of sample, and you can see here in this picture, I start with this. Oh, when I come, oh, it's not so complicated, it's easier. And after I start to dilute the 300 sample in a day, and of course she helped me a lot. This is my table now. I put this picture in this morning of the table. Hey, look, now I look my table right like in this picture, because I have a lot of work to finish a part of my thesis, and to have a lot of good results. A part of this result are presenting in this conference, in the previous section. And of course, I have a lot of things to speak about the people from here, because we have expression in Romania. I don't know to translate this in English, so I take this expression in Romania. The expression is omul sfințește locul. So that means here, in my opinion, here the people are important, and when all this period I see all the support for the Professor Valente and for my colleagues. They help me to understand what the things are wrote in the lab, and of course in the outside, because sometimes after the lab we go to drink a juice or a coffee to talk more about our other things about our life, not about the lab stuff. So I'm very glad I have this opportunity with the Erasmus and mobility grant from NeXT-Chem to come here and to have this good result, and to have this experience in my life, because I think it's a unique experience to go in another country for some months, to stay and to work in another country, to come back with results and a new vision, compared with your country. So the culture here in Portugal, it's very interesting. They have a lot of interesting things, and they eat a lot of fish. And honestly, the fish, it's not my favorite dishes, but after 3 months, I start to understand and to eat fishes with pleasure. So I think Portugal, it's a country when you need to go to see all the beautiful things that are here. And thank you for your attention.
Thank you, Andrea, too. The very nice presentation and very true. If there are questions from the room or from online? If not, I think that now you understand that we can use this tool, Erasmus grants, to improve the knowledge of the young researcher in our institute. And I invite also our colleagues to make this experience, and they will have very good returns about such action. So thank you very much, Andrea. Thank you to all for this nice presentation about your experiences. And now I invite for the last part of our workshop is the last presentation. It is publishing opportunities, how to increase your chances of publishing, which is presented by Rareș Vasilică from e-information. Please. Da, poate fi în română, poate fi în... Deci aveți maxim 30 de minute. Așa, vă rog frumos că suntem cam întârziați.
Așa. Bună ziua tuturor! Mulțumesc pentru invitație. Scopul prezentării de astăzi noi am denumit oportunități de publicare cu M-Formation. O să vedeți câteva servicii pe care M-Formation le pune la dispoziție mediului academic românesc. O să vedeți că sunt undeva la 5 servicii pe care noi le promovăm continuu. Cel mai popular, să spun așa, în momentul de față este profilul instituțional. Bănuiesc că mulți dintre dumneavoastră deja beneficiați de accesul la bazele de date și aici mă refer la Web of Science, o platformă care vă oferă informații statistice, Scopus, Google Scholar, care la fel poate fi accesat, doar că în cazul acesta e gratuit și, bineînțeles, platformele full text, de unde puteți să extrageți informații și aici mă refer la cărți, reviste și articole. Practic, asta este una din multiplele variante pe care le puteți accesa prin profilul instituțional. Mai departe o să vedeți portofoliul de servicii. Nu sunt neapărat niște servicii, le-am denumit așa ca să fie un pic mai ușor de înțeles și aici aveți prezentat masterclass, academic publishing services, servicii profesionale de editare în parteneriat cu Enago, Integrity și revista pe care eu și colegii mei vă punem la dispoziție și anume Research and Education. Așa. O să începem cu Academic Publishing Services. Information, în momentul de față, este o companie care are peste 25 de ani de experiență în tot ce înseamnă partea de furnizare a resurselor electronice. Vine în ajutorul mediului academic pentru publicare. În momentul de față, noi colaborăm cu cele mai utilizate și prestigioase edituri și anume Springer Link-ul, De Gruyter, COL și bineînțeles putem să facilităm partea asta de comunicare și cu alte edituri care nu sunt neapărat menționate. Aici aveți un scurt calendar, să spun așa. Pare foarte lung pentru că sunt foarte multe informații, dar noi am încercat să le scurtăm. Practic aici aveți un calendar cam cât durează tot procesul de publicare pentru un tânăr cercetător și nu numai. O să vedeți că perioada întreagă poate să ajungă undeva între 6 și 8 luni, asta dacă toate lucrurile funcționează perfect, adică dacă nu există varianta în care lipsesc anumite lucruri, dacă documentul nu corespunde și așa mai departe. Tocmai de asta Academic Publishing Services vine în ajutorul dumneavoastră ca să putem să punem la dispoziție o variantă complexă pentru documentele dumneavoastră, astfel încât perioada pe care o vedeți aici de 6-8 luni chiar să se respecte și să nu ajungem la un an, poate chiar mai mult de atât. Aici aveți câteva variante prin care noi le-am publicat în colaborare cu Elsevier și COL, câteva cărți și câteva reviste. Iar în continuare aș vrea să vorbesc un pic despre serviciul Integrity, care este în parteneriat cu Turnitin. Turnitin în momentul de față colaborează cu marile universități la nivel mondial. Integrity este o platformă care verifică similitudinea, practic puteți să vedeți nivelul de plagiatură al unui document, dar că nu este o platformă este o platformă foarte, foarte complexă. Poate identifica inclusiv varianta în care un document este preluat în engleză, tradus în română și apoi din română tradus în italiană. O să știe exact site-ul, de unde a fost preluat, sursa și toate lucrurile de genul acesta. Pentru accesarea serviciului Integrity, puteți să faceți lucrul ăsta de pe pagina Information, imediat lângă profilul instituțional. O să puteți beneficia de această unealtă. Aici aveți o scurtă prezentare, o să vedeți că platforma este foarte simplu de utilizat. Trebuie să denumiți proiectul dumneavoastră, vă alegeți fișierul din computerul dumneavoastră și îl trimiteți către platformă pentru a fi verificat. Încă un avantaj pe care îl are Integrity este faptul că vă oferă rezultatul, practic vă generează un raport și vă oferă rezultatul în câteva secunde. Există foarte multe programe antiplagiat, doar că în momentul în care devin un pic mai aglomerate, durata în care puteți să vedeți raportul respectiv poate fi de ore, zile sau chiar săptămâni, mai ales dacă vorbim de un program antiplagiat la nivelul universităților. Acolo se poate duce și către o săptămână să vă oferă un rezultat, mai ales în perioada de sesiune. Serviciul profesional de editare în parteneriat cu Enago complică și mai mult lucrurile, să spun așa. E o platformă foarte complexă, unde puteți să încărcați un document care nu este neapărat finalizat, practic o cercetare sau un alt tip de manuscris, spre exemplu, și această platformă, practic, poate să vă ofere servicii de editare și traducere. Spre exemplu, dacă vreți să publicați cu o editură din Statele Unite, automat Enago o să vă pună în contact cu o persoană care se ocupă de genul ăsta de cercetare. Practic, fiecare editor din interiorul echipei Enago o să meargă direct pe subiectul dumneavoastră. E foarte important să aveți în vedere că tipul de engleză pe care trebuie să-l utilizați este cel american, pentru că dacă o să utilizați engleza britanică, este foarte mare posibilitatea ca documentul dumneavoastră să fie respins. Asta se întâmplă în ambele situații. Când publicăm cu edituri din Marea Britanie, folosim engleza britanică, când publicăm în Statele Unite cu engleza americană. Și aici e foarte important să apelați, dacă nu o să apelați la genul ăsta de serviciu, e foarte important să apelați la traducători care se ocupă de engleza academică. E foarte important lucrul ăsta pentru că un traducător de limbă engleză de multe ori nu o să poată să meargă la standardele unei edituri. Mai departe, unul din serviciile preferate de mine este masterclass-ul. Acest masterclass este gândit astfel încât să punem la dispoziție 5 cursuri, 5 module le-am denumit noi, tocmai ca să putem trece prin tot procesul de publicare. Aici, împreună cu traineri dedicați pe subiectele de publicare, o să puteți să beneficiați de suportul nostru, o să facem tot timpul, cum să le spun, chestiune aplicată. Deci, întotdeauna în momentul în care participați la un astfel de modul, noi o să preluăm documentele pe care dumneavoastră urmează să le publicați și o să vă oferim feedback în funcție de situație. În momentul de față am mers pe o variantă de maxim 15 persoane, tocmai ca comunicarea între trainer și participanți să fie una cât mai benefică. În momentul în care există mai mult de 15 persoane, de obicei e destul de complicat în a comunica și ca timp estimat pentru fiecare dintre aceste 5 module este de 5 ore și 30 de minute. Noi am aproximat toate lucrurile astea, vă dați seama că nu o să fie la 5 ore și 30 de minute, am oprit totul, toată lumea merge acasă și nu, sunt doar niște lucruri aproximate. Cam astea sunt cele 5 servicii, dacă aveți întrebări pentru ele. Nu? Ok. Dacă nu sunt întrebări, pe online bănuiesc că nu avem cum să le... E perfect. Ok. Dacă cineva de pe online dorește să adreseze o întrebare.
Mă auziți?
Da, sigur.
Vă auziți. Deci, haideți că este, pentru că tot este pe internet și la noi și a și discutat cu OPE anul trecut situația asta. Pentru cei care nu știu, COPE este o asociație a editorilor de reviste științifice. Este o mare problemă cu papers mill și cu autoratul nejustificat, cu autoratul cumpărat. Acum, cred că ar trebui pentru că colegii noștri mai tineri au tot primit și trebuie să facem clar o diferență între ceea ce înseamnă paper mills și ceea ce... Mă auziți?
Da, vă aud un pic încet.
Deci, mai tare, eu zbier foarte tare, e povestea asta. Deci, încă o dată, rugămintea e să facem o diferență foarte clară între ceea ce înseamnă paper mills. Paper mills sunt niște firme care îți fac și ele de articole, dar nu ți le editează pe ale tale, ei ți le fac.
Da, Enago nu are legătură cu ce îmi spuneți dumneavoastră. Practic, partea de cercetare și tot ce ține de document o să trebuiască să vină din partea dumneavoastră. Noi nu ne ocupăm să facem cercetarea în locul dumneavoastră.
Exact. Aici vreau să subliniez această diferență semnificativă. E o diferență semnificativă, înțelegeți, ca să priceapă, pentru că primesc tot felul de colegi, mai ales colegii tineri, primesc tot felul de mail-uri și nu știu de ce Lituania, datorită probabil sistemului dezvoltat de la ei. Din Lituania sunt majoritatea, în care de fapt sunt astfel de companii care și generează conținutul. Deci să facem această diferență între editare de conținut științific care ne aparține nouă și a avea articole la care devii coautor în paper mill. Aceasta vreau eu să subliniez.
Da, e foarte bine ce ați spus dumneavoastră acum și mulțumesc pentru lucrul ăsta. Mi-am adus aminte acum de o doamnă, apropo de firmele din Lituania și de prin altă parte. Am avut un mail la un moment dat, o doamnă care a trimis la o firmă din Spania, sau nu mai aduc aminte, a trimis un articol, a plătit undeva la 400 și un pic de euro și era foarte nemulțumită de faptul că revista respectivă nu i-a publicat documentul și de ce nu apare în Web of Science și toate lucruri de genul ăsta. Dacă aveți genul ăsta de e-mail-uri, dacă cineva vă contactează și vă cere bani pentru publicare, măsurați de foarte, foarte multe ori, pentru că e posibil ca revista respectivă să aibă un website absolut minunat în care apare factor de impact 50, cea mai bună, cea mai frumoasă, și când începeți să căutați informații, o să vedeți că nici măcar nu e prin top 100 sau ceva de genul ăsta. Deci oricine poate să promoveze o revistă ca fiind perfectă, să vă ceară bani și automat o să-i pierdeți, pentru că nu aveți nicio șansă să puteți să recuperați banii, pentru că deja s-au oferit niște servicii. Nu neapărat să publicați să apară documentul dumneavoastră în Web of Science sau în Scopus, dar deja s-au făcut niște lucruri. Deci evitați pe cât posibil și încă o dată mulțumesc pentru sesizarea respectivă. E foarte important să aveți în vedere că serviciile pe care noi le punem la dispoziție sunt pentru, vin în ajutorul dumneavoastră, nici într-un caz să facem partea de cercetare. Pentru mine, ce s-a prezentat astăzi, chiar să stăm și mă uitam, e o altă limbă, adică chimia nu sunt doar niște numere, A, B, ok, mai știu, carbon și alte lucruri. Dar nu facem partea de cercetare pentru dumneavoastră. Noi cu ce putem să vă ajutăm este efectiv procesul ăsta complicat, aș spune eu, pentru că durata de 6-8 luni nu este una foarte, foarte mică în varianta asta de publicare. Mulțumesc încă o dată pentru întrebare și dacă mai sunt întrebări, o să vă rog să mi le adresați.
If there are any question, please. So if not, we take a small break. very short because we are 5 minutes, let's say, and we are coming here for the award ceremony. Yes, this is the next step. Thank you.